It's a great pleasure to basically give a general background introduction to what Victoria is going to talk about in the second, more detailed part. And what I'll basically try to do is focus indeed on the big picture of how can we use computational omics to decipher the chemical language of the microbiome. What is the chemical language of the microbiome? Well, of course, in microbiomes, in animal microbiomes, plant microbiomes, human microbiomes, we see that there's a lot of interactions going on between bacteria and the host, between beneficials and pathogens, etc. And many of these interactions are actually mediated by molecules. Very frequently specialized metabolites, but sometimes also like primary metabolites. Here you see some examples in the context of a plant microbiome, where we know that, for example, pathogens may produce molecules like pathogenicity factors that actually facilitate the infection process. There may be other bacteria that actually inhibit the pathogens by making molecules that are actually, for example, antimicrobial or antifungal. There's also interactions between the microbes, and sometimes also the host actually interacts with the microbiome itself through such metabolites. And these metabolites, you could consider them as the chemical language of the microbiome, and they fulfill all sorts of roles. So traditionally, many people mainly thought of like antibiotics as the key kind of function of these kinds of metabolites. And of course, there are many antibiotics and antimicrobials out there that do play a role in microbiome interactions. But I think recently it's become much more clear that the whole range of different functions of these molecules is also much broader. Of course, it's well known that you have molecules involved in communication, like quorum sensing signals, for example, but also, for example, molecules that had traditionally been thought of as toxins, like doxorubicin is a cytotoxic molecule that is actually an anti-cancer drug, was recently shown to actually play a key role in phage defense. So the ecological role may actually be quite different from the biological activity that was initially measured. Similarly, there's many molecules that are involved in nutrient acquisition. You see here also at the bottom right, you see phenazines, which were traditionally known as kind of weak antibiotics. But Diane Newman's group from Caltech actually showed that there are actually redox active molecules that play a role in mobilizing, solubilizing nutrients like phosphorus. So lots of different roles that all of these molecules have, and that also explains why they're so incredibly diverse. So actually, when you look across bacteria in microbiomes, you see that many bacteria that have basically the same 16S sequence, that are part of the same species, these bacterial strains can sometimes have vastly different metabolic capacities with regard to these specialized metabolic functions. So they may make quite different molecules. So we can also see this by looking at the way the production of these molecules is encoded in genomes. For example, for secondary metabolites, we look at what we call biosynthetic gene clusters. So these are clustered groups of genes that are adjacent on the chromosome and that encode for all the enzymatic machinery to make these molecules, also to transport them out of the cell, etc. And you see often there's a correspondence between the chemistry of these molecules as well as the genes that are part of these gene clusters. Here you see, for example, this moiety in blue, coumarin, moiety, and rubidirin is shared with this other molecule in semocyclinone. And you see also between these gene clusters, the corresponding genes that are responsible for making that part of the structure are also shared. So looking at the genes within these gene clusters can therefore tell you quite a bit about the chemistry. And this also tells us, if we look at the genomes of these organisms, that indeed, very frequently these gene clusters are gained or lost within species, or they're horizontally transferred, and that actually leads to a lot of ecological specialization. And because of this, actually the whole repertoire of these molecules is extremely diverse. So there's really many hundreds of thousands or maybe millions of different bacterial molecules out there. And we recently teamed up with the group of Nadine Siemert in Tübingen to basically do an analysis for these secondary metabolite biosynthetic gene clusters of how diverse are they really, and what percentage of all these molecules do we actually already know. And that was done by basically identifying across publicly available genomes all of these biosynthetic gene clusters, then grouping them into families through clustering algorithms. And actually, we use two independently developed clustering algorithms, one in my group, Big Slice, and one from the Helmholtz Institute of Pharmaceutical Research in Germany called Clustomatic. But as you see, they actually gave similar results. These were used to group the gene cluster family, to group these gene clusters into families in a way that ideally all of the gene clusters in one family correspond to pathways that are making similar molecules. And we did that at a level of granularity that was calibrated or fine-tuned based on a database of chemical structure called the Natural Product Atlas, so that in cases where for a gene cluster the chemical product was known, we made sure that gene clusters with chemical products that are part of the same chemical classes in NP Atlas would also end up in the same family. So in that sense, gene clusters were clustered at the same level of granularity that the molecules were clustered in NP Atlas as chemical classes. So if we then do a rarefaction analysis, we can predict how many of these chemical classes are out there in the biosphere based on currently available genomic information. And according to this analysis, you see that you end up with a prediction of, well, between 70,000 and 100,000 different chemical classes, of which at the moment only 2,500 are known. And that equals around 3% of the predicted chemical space as being known. Yes.
Can I ask in this context for the MACs, what environment are they covering, and was there some quality filtering involved, so were there only high quality ones or also lower quality MACs? So what was the genome assemblage quality of those?
Yeah, so the analysis on the left, that was based on only isolate genomes. And then, of course, we also checked, and that's the analysis on the right, like if we look into metagenome assembled genomes, that may cover also part of the uncultivated majority of microbes. Does that actually change the situation? And of course, it's a bit difficult with MAGs because actually very frequently these gene clusters are missed in MAGs because they are often strain specific. And if you have a combination of strains in the same metagenomes, they will not be correlated in terms of abundance with other contigs of the same genomes. But yeah, if we looked here at several high quality MAG collections that were available at the JGI, and that's what Satria did, the person who worked on this. And yeah, basically what he saw is that, of course, in terms of known types of gene clusters that are in this database, MyBig, and I will tell a little bit more about that later, of known biosynthetic gene clusters with known products, there's very limited overlap with metagenome assembled genomes. And there's also quite a bit of, quite a substantial fraction of gene cluster families that are only found in the metagenome assembled genomes and not in any isolate genomes, which would actually indicate that maybe we know less than 3% of everything that's out there. Yes? So then the question becomes, like, if indeed it's really the vast majority of the microbial chemistry in these microbiomes and in the microbial biosphere that is yet unknown, is how do we mine that diversity? How do we figure out across these millions of gene clusters and these tens of thousands of chemical classes that are likely out there, which are the ones that really matter? Because of course we can't go after them one by one, because, well, actually structurally characterizing one of these molecules, elucidating the biosynthetic pathways, that's at least like multiple PhD projects. And if you multiply that by tens of thousands, you can kind of calculate that's a few centuries of work likely still with current biochemical technologies. But we would really like to find out, like, where are the hidden gems inside this biochemical diversity. And this is a picture that was made by an artist from Nature Reviews Genetics when we had a review there, and it kind of nicely shows the process of what we call genome mining. So we have the DNA sequence here that's going in this mine shaft, and it's lined by a lot of stuff, but only some of that stuff are the hidden gems. And what will be the hidden gems? And I would say there will be two things, two general categories that I would see as important things to go after. One of them is how to find those molecules that can really explain mechanistically and functionally key microbiome-associated phenotypes, such as phenotypes associated with health and disease. And second, of course, also molecules that could lead to, for example, the development of new drugs or new crop protection agents or other molecules of societal relevance. So in this process of mining the biochemical diversity of the microbiome, I will lead you through three steps in terms of the algorithms and tools that we use for this. First of all, it's the identification of these gene clusters. Second is how do we chart their diversity across large data sets of many genomes or metagenomes. And thirdly, how do we start predicting functions? And that really boils down to prioritizing that large diversity to the specific gene clusters and molecules that really matter. So let's start at the start, like the identification of these metabolic gene clusters. And this is something I actually started with when I was a PhD student myself, a little bit over 15 years ago, when there were just a few genomes available. And I was given one Streptomyces genome myself to look for biosynthetic gene clusters. And I was doing it some kind of semi-manually by doing like blast searches and these kinds of things. And I found it very tedious and boring. And I thought, okay, it should be possible to make this more automated. And actually at the time, there was a project here at the Broad Institute where people were sequencing like 20 genomes. And back in those days, like 20 genomes was like, how do you even start analyzing that, right? That's just impossible. But we did start working on it in collaboration with a group in Germany, Kai Blin and Tilman Weber. And we basically used a general strategy that is still being used in many of the tools today, simply because it really allows a very precise tuning of the identification of these gene clusters, where you start with an input genome sequence, and then you use a rule-based approach. It's basically by finding specific combinations of protein domains that are co-located adjacently in the same chromosomal region, on the same chromosomal locus within a defined distance. And that specific combination of domains is indicative of the presence of a certain type of biosynthetic gene clusters, because these domains represent like the key enzymatic domains that catalyze the key reactions for such a type of pathway. For example, here we have like 3 key domains for a given pathway types, A, B, and C. And you might then create a rule that says, okay, if we find either or domain A with either domain B or domain C, then we're going to trigger the detection rule saying, okay, this must be this and this kind of pathway. And these kind of logical rules you can tune yourself and also fine-tune, calibrate in a lot of detail. And in a relatively complex space, it really allows you to manually kind of draw a line around your data points, so to say, to very accurately predict well-known types of biosynthetic gene clusters. Of course, this may not be the best approach for finding entirely novel kinds of pathways, but actually, when we look at most of these types of gene clusters, the core enzymology is very well defined, and that actually makes this generally more accurate than, for example, machine learning methods, which are very useful for finding novel things, but not so much for finding these well-known types of gene clusters based on relatively small amounts of training data. So this is formalized in...
Hi.
Yeah.
Can I ask a question, please? With these rules, like, how often do you, I don't know, this is kind of a vague question, but if you're just looking within a genome sequence and you have these rules versus when you're looking in a metagenome, and it might be that A and B are in one bug and C is in the other, and now would that be considered passing the rule, or is this all just within a genome sequence?
This is all within one chromosomal locus, within one region in the chromosome. So generally, the assumption is that all of the genes are clustered in the same genome, in the same genomic location. And that is in bacteria for these kinds of pathways, it's usually the case. And
So
in
they don't ever get
95
just,
to
like,
99% of the
split
cases.
to another part of the genome, like A and B is here and C moved somewhere, like, I don't know, a million bases away or something. That's rare.
That's very rare in
Okay.
bacteria. So actually, evolution somehow tends to optimize for the genes to be very tightly clustered, probably because they're co-regulated in this way. They move together because there is also a lot of horizontal gene transfer. And there's a couple of reasons why this is likely the case. If you look at other organisms like plants, actually, for example, like maybe half of the biosynthetic pathways are not clustered in plants. And there we actually need to rely on other things like co-expression to connect these genes. And you can't just rely on the co-localization of the genes. So this anti-smash software, which I started working on as a PhD student, is well, it actually has undergone many versions now. We're about to release version 8, where not only does it identify the biosynthetic gene clusters, but there are also a range of different follow-up analyses to kind of annotate the gene clusters in detail in terms of the enzymes encoded, comparative genomics, et cetera, et cetera. So I was just saying that for most of the biosynthetic pathway types, we can actually very accurately detect them in this way. I think for the majority of the canonical secondary metabolic pathways and gene clusters, we anti-smash detects them like with more than 99% accuracy. There are also some kinds of pathways that are actually more tricky and more difficult, and that's because they don't work very well with this kind of rule-based concept, because there is not domains A, B, or C that are always present. And in particular, this is the case for the ribosomally synthesized and post-translationally modified peptides, which also often play key roles in microbiomes. These are also called RIPs for short. So that's the abbreviation. And the thing is, how these gene clusters are put together is you have this tiny precursor peptide encoding gene, like a gene that encodes for the production of a small peptide. And then you have some transporter, often to transport it out of the cell, some modifying enzymes, but they could actually be anything. So that actually makes them really diverse. So there's not, of course, within specific classes of these RIPs, you find the same modifying enzymes. But it's becoming clear that there's really many dozens or maybe hundreds of these classes out there that all use different modifying enzymes. So there is very little in common in terms of protein domains between like the full breadth of these types of gene clusters. And that makes these specific types of gene clusters actually more challenging to detect. Yeah, so these modifying enzymes, they could be anything. And also for this reason, we actually came up with a specific like alternative way of approaching this to find these types of gene clusters, where we actually start with a machine learning model that first tries to identify these precursor peptide encoding genes. It's trained based on all of the known precursor peptide encoding genes, and actually you see that there is a number of physical chemical properties related to their amino acid composition, charge characteristics, and others that actually sets them apart from other small genes. So there are certain physical chemical characteristics that these peptides, they tend to have in order to be stable, not to be proteola degraded very quickly, and also to be bioactive. And in that way, we can actually separate relatively well potential precursor encoding genes from other small genes. But of course, there's so many small genes out there that even if you can do this at 99% accuracy, you'll get still a lot of false positives. So we had to also combine that with other criteria. And we do that by also looking like, are these putative precursor peptide encoding genes, are they located in an operon structure with other genes that are enzyme encoding? And we're not limiting ourselves in this case to specific enzyme families, but we just want to see enzymes, because that indicates that there's something enzymatic going on. And another criterion is that many of these gene clusters actually are flying in and out of genomes. So they're usually not part of the core genome of a species or a genus. So that's why we can also measure that with like an orthology conservation score, saying if they're part of the core of a pangenome of a species or genus, then they're much less likely to be RIPs, because these are often specialized functions that are strain or species specific. If they're part of the accessory genomes, they're much more likely to be RIPs. And if we combine basically all these criteria and then we dereplicate it by removing all of the stuff that we can already identify with anti-smash, that actually gives us a data set that is actually small enough to manually go through and highlight basically candidate new classes of RIP biosynthetic gene clusters. And we did this a couple of years ago in Streptomyces, which is a model bacterium for secondary metabolism. And already there in this one genome, in this one genus, we found around 40 to 50 candidate new classes of RIP biosynthetic gene clusters. And that corresponds to actually thousands of biosynthetic gene clusters in these genomes. And by now, we've also run this on a range of other genera, including genera from the human microbiome. And there is really a lot to discover in this way still. So that's pretty exciting.
Can I also ask another question? Is the ordering of genes important, and also what strand the gene is on? So can I basically have a biosynthetic gene cluster with genes on different strands, and would that be considered somehow?
Yeah, so sometimes biosynthetic gene clusters can be pretty large, like some of the largest ones can be 100 or 200 kilobases, and then you might have 10 or 20 operons that are pointing in different directions, but they're still clustered. So yes, that's totally possible. The order of the genes, I don't think there's actually been a good paper to quantitatively prove this, but actually quite frequently the order also matches the order of reactions. Not always, but more frequently than not. And this probably has to do with like being able to quickly make molecules that the enzymes that carry out the first steps need to be synthesized first. They start carrying out the first reaction, and then in the meantime, the next proteins are also made, etc.
Can I ask another general question?
Yes.
Like if you look in a bacterial species where like more is known, like E. coli or something like that, and you try to, I don't know, identify the fraction of the genome that's biosynthetic gene clusters or that makes RIPs, like what is that fraction? And then like how does that fraction compare to just like the average genome that you take, I don't know, say in an environmental metagenome sample and you try to like annotate things, like how much does like a really well-studied genome where we can annotate everything compare to one where we know much less? Because I'm just trying to like, so all of this is, I don't know, at a high level kind of based around homology. Is that kind of right? Yeah. So I'm just kind of curious, like how much more do we need to know for an environmental sample, do you think?
Most of these enzymatic pathways are actually pretty ancient. So it's really the same families of enzymes that you find like in model organisms versus uncultivated ones. Although I am sure that there will be stuff that is very like specific to a specific clade, but then these are usually also less widespread types of pathways. So there's stuff that we're missing probably in this sense. But if we look at like the widespread general classes of biosynthetic pathways, I don't think there's a big difference in our ability to annotate them in E. coli versus like uncultivated acetobacteria or something.
Helpful, thank you.
And like the percentage of the genome devoted to it, it can range from like 1% to 20%, or yeah, in that order of magnitude. Yeah. So then at some point we also became interested, and this was in a collaboration with Michael Fischbach, who was currently at Stanford University, and I spent some time in his lab about 15 years ago when I was a PhD student, and he basically came to me and said, well, actually in the gut microbiome, there's so many of these primary metabolic gene clusters that are also frequently strain or species specific and really drive the ecological niches of the bacteria that harbor these types of gene clusters. For example, you have gene clusters for converting primary to secondary bile acids. There are gene clusters for short-chain fatty acids production, amino acid catabolism, et cetera, et cetera. So yeah, that's basically what started the project that Victoria will tell more about, where we made like another version of or flavor of anti-smash, which was called gut smash, which was really focused on finding all of the well-known types of catabolic gene clusters and their pathways that are also relevant to gut microbiome research. Yeah, Victoria will tell a lot more about that in her talk. In the meantime, we're also working on other flavors. For example, we've been recently building, and that's about to come out, Rhizosmash, which is for plant microbiomes, where we actually look at catabolic gene clusters for the catabolism of root exudates, because like plants, they exude 20% of their photosynthetic carbon to the rhizosphere. So actually a lot of organic acids, sugars, et cetera, they're exuded to actually recruit and modulate their microbiome. So understanding like what kind of catabolic pathways and gene clusters the members of the microbiome has, and in this case also tell us like how can plants recruit which microbes to their microbiome. Okay. That brings me to the next step, and that is really charting the diversity of these gene clusters, because of course you can identify them in single genomes, but of course, yeah, generally we don't work with single genomes, not even with 20 genomes like in 2010, but maybe 20,000 or 20 million genomes. And for that, we actually, together with international collaborators, build an extensive software ecosystem of tools that are interoperable with each other, that really allow this kind of large-scale analysis. I already mentioned briefly the big slice clustering algorithm at the start, which groups gene clusters into families. In general, we start with the gene clusters that we can predict or identify in genomes with anti-smash, gut smash, rhizosmash. Then we have databases like MyBig for known biosynthetic gene clusters, which at the moment hosts about 3,000 gene clusters with known chemical products. And then we can start grouping these gene clusters into families with either with sequence similarity networking, that's in Bigscape, or with vector-based clustering approaches like in Big Slice. When you do it with Bigscape, you get these networks where each node represents one biosynthetic gene cluster or metabolic gene cluster, and they're connected to each other if their mutual similarity based on a number of distance metrics, and I'll come back to that in a moment, is higher than a certain threshold. And in this way, you can, with graph clustering, also group them into families to know which gene clusters across a set of genomes are actually closely related to each other and are likely to encode for the same kind of pathway. So coming back briefly to this database of known biosynthetic gene clusters, this is also really fun project, which has really been an open science initiative. So earlier this year, we actually ran like massive online parallel annotation marathons. So we had like 6 half-day Zoom sessions with more than 250 people who all came online to actually annotate all of these gene clusters in a standardized way. They're subdivided into teams based on their expertise. We had a review system to actually ensure also the quality of the annotations. And in this way, we can really get a lot of information that is out there in the literature into this database in a standardized format. And it's really also the basis for a lot of the follow-up like prediction algorithms that you can build within this space. So then to Bigscape, the clustering of these gene cluster families, the key trick that we employ there is that we start by basically translating the sequences of these gene clusters to strings of protein domains to kind of have a small scale representation of a gene cluster. So you do protein domain calling, and then you have, let's say, strings of protein domains that represent a biosynthetic gene cluster. And in the next step, these gene cluster representations, they can be compared to each other using a couple of different metrics. The simplest metric is just to say, okay, Jacquard index of the protein domains. Like if you have a pair of these gene cluster protein domain strings, what fraction of the protein domains are shared between both of them or are unique to either one or to the other. You can also do that with pairs of adjacent protein domains, which tells us something about the conservation of order. And that can be done with also with triplets, for example. And then finally, but also importantly, we want to have some indication of how similar these domains actually are to each other in terms of sequence, because some of these protein domains can have very broad functions, and you actually need to know how closely related are they really in sequence, because that tends to correlate pretty well with the chemistry and the catalytic activity of the enzymes, of course. So this is done. Sometimes you have different copy numbers of the domains, and that also needs to be taken into account. So yeah, in case there's like 2 domains in one gene cluster and 1 domain in the other gene cluster, we'd regard like the best scoring pair of these to take the sequence identity between them, and the other we would basically give a 0, so that the duplication index is also taken into account. And then for the sequence identity itself, of course, these sequences need to be aligned, and actually by far the quickest way to do that is to align them all against the profile hidden Markov models with which they were detected at first. So initially we did it using multiple sequence alignment, like muscle and MAF type of sequence alignment, but that scales horribly because you have all versus all comparison, and it basically takes a lot of time. But by actually aligning all the sequences against the hidden Markov model profiles, actually it's almost linear in terms of runtime when you go to larger samples. So that makes a big difference. And then we can visualize this, of course. So here's an output of first version of Bigscape, where you get this kind of output for, in this case, 120 genomes. You see like an absence-presence matrix of families of gene clusters across different genomes. And then also go to a specific type of gene cluster, like in this case, a type of pathways related to PKS, polyketide synthases. And here you can interactively really also browse and explore these kinds of network, for example, also by typing in keywords like the molecules that are related to these known biosynthetic gene clusters, reference BGCs from MyBig, and it will highlight it in the network, and you will immediately see which other gene clusters from certain organisms are closely related to this, and you get a multi-locus phylogeny also of these gene clusters to see how they're related and what are the key similarities and differences. At the moment, we're bringing out a second version of this, which is going to be a lot faster, about 10 times faster through a number of additional tricks, and also through local storage of all the intermediate data. You can actually use this to hundreds of thousands of gene clusters easily. Yeah, basically without RAM limitations. Okay, I'll briefly skip this because I want to have a little bit of time to go to the last part of my talk, which is a bit of the forward-looking part, which is how do we really predict which of these gene clusters actually matter? What are their likely functions? And I think this is really one of the key questions in this very large genomic space. So there's so many of these metabolic gene clusters out there, which have all sorts of functions. And we really need to predict, for example, in a microbiome setting, which ones really matter to explain a certain microbiome-associated phenotypes. And this is really a key challenge that I've been tackling with my group as part of an ERC grant that is finishing next year already. It's called the Decipher, where we've been following a couple of different strategies to try to predict functions of these biosynthetic gene clusters. A first strategy that we've been following is actually expression-based. So sometimes you have like a range of samples and you have phenotypic metadata on them. For example, health disease metadata, and you can actually look at, for example, the expression of a BGC and see is that associated with the phenotype or not, very simplistically. And in this case, if you have the microbiome-associated phenotype here at the left across samples, and you see that the expression of this red gene cluster corresponds much better to the phenotype than the expression of the blue gene cluster. So yeah, from that you might think, okay, maybe the red gene cluster is more likely to be involved in mediating that phenotype. But what you could also do is actually use the expression patterns of other genes and other gene clusters, either within the same genome or across genomes of different members of the microbiome, and use these as your phenotypes. So whenever a certain gene cluster is expressed, what happens in the rest of the cell? What happens in the rest of the microbiome? Could that be causally related, potentially? And for this, Zach Wright, former postdoc in my lab, developed a tool called CocoLocus, which generates these co-expression networks of metabolic gene clusters and other genes in the genome. Here, the large nodes are the gene clusters in a given genome, and they're represented by, say, an eigenvector of the expression levels of their constituent genes. And all the gray smaller nodes, there are other genes in the genome that are strongly correlated in expression with at least one of these metabolic gene clusters. And you can also already see that there's groups forming of gene clusters that tend to be co-expressed. And you can also dive into that further by also including regulatory information, for example, transcription factor binding sites. And here you see, for example, and this is work of Hannah, who is down here, who actually annotated this network and saw that like all of the genes and the gene clusters over here, they all have binding sites for this specific regulator called DMDR1, which is involved in iron response. And actually, two of these three gene clusters, two of these three nodes over there, they're actually encoding the biosynthesis of known siderophores, which are iron scavenging molecules, which also play a key role in like the battle for iron between microbes and the host and the microbiome. But at the same time, she also saw another gene cluster in that same part of the expression network, of the co-expression network, which was an unknown gene cluster for which we didn't know a function yet. And she made CRISPR-Cas knockouts of all of the genes in that gene cluster, and it actually turned out to be also involved in siderophore biosynthesis, which really indicates that indeed you can use this kind of inference to predict functions for a gene cluster. Specifically, this gene cluster, which you see over here, it's the top gene cluster, is working together with one of the other gene clusters to produce a specific subclass of these well-known siderophores called desferrioxamine, namely the acid-capped desferrioxamines. And actually, when you knock out these gene clusters, then you see that there's no effective production of these siderophores. And I think what is really cool is that not only can we predict the function of a gene cluster, but this was also in a model organism, Streptomyces coelicolor, which has been studied in terms of its biosynthetic gene clusters for decades. And this specific pathway for desferrioxamines is also a very well-known model pathway for siderophores, which people also have studied for a long time already. And still, nobody had ever known or hypothesized any role for this particular gene cluster. And based on the combination of co-expression and co-regulation, we could really figure out this function. So I think this is a promising strategy to figure out what these kinds of gene clusters are doing. I also mentioned like cross-species correlations and how they can be helpful. And Robert Koetsier in my lab is pursuing that further. He has been trying this out with a number of model synthetic microbial communities, and one of them is a synthetic community from that's a model for a rhizosphere microbiome. It's composed of these 3 bacteria: Pseudomonas coriensis, Flavobacterium johnsoni, and Bacillus cereus. And when you actually look across a whole range of co-cultures, like both dual co-cultures, triple co-cultures, mutants in different conditions, etc., you actually see that there's also specific cross-species correlation and anti-correlation in the expression of the metabolic gene clusters. For example, this Pseudomonas has a gene cluster for a molecule called coriensin, and this is an antibiotic. And whenever Pseudomonas makes that antibiotic, it seems that the Bacillus strain actually responds by turning off all its siderophore production. But it's also highly positively correlated with these 2 gene clusters over here, which actually encode the production of molecules that are known to be protective against redox stress and also against redox stress inducing antibiotics, so called bacillophiol. So whenever the antibiotic is produced by Pseudomonas, Bacillus will start to basically produce these protective compounds, and at the same time, it also increases the expression of efflux pumps and other kind of defense mechanisms. And in this way, we're thinking that this can also be a way, for example, to find antibiotics. So whenever a certain one bacterium in a microbial community expresses all of these defense mechanisms, it's likely that there's another member of the community at the same time that is producing some kind of antibiotic. And by looking at the cross-species correlations of expression, it should be possible to also hopefully, that's the next step that we're going to do in more complex communities, to unravel these antagonistic interactions between microbes and the genes and gene clusters that are mediating them. And finally, one other strategy that we're using to predict function for these metabolic gene clusters is basically through prediction of the structures. Of course, there's many databases of bacterial metabolites and their chemical structures, and often also associated with functional metadata like biological activities. We know that, for example, beta-lactam rings are very often associated with antibiotics. You have these chelating moieties that are binding metals that are often associated with metallophores or specifically siderophores, etc. So we can find association of substructures of molecules with specific types of biological activities. At the same time, we can actually predict also structural features from gene clusters by, for example, subset specificity predictions of enzymes, or by finding these subclusters, these groups of co-evolving genes within gene clusters that are associated with the biosynthesis of a specific chemical moiety. And thus we can also get predicted structural features from the gene clusters. And then we can see, is there a match between, let's say, the predicted structural features from gene clusters and association of these structural features with certain types of activities of interest. And as an example, Zach, whom I mentioned earlier, actually formalized this for metallophores. So these are molecules that are metal binding by, in detail, looking at all of the sub-pathways within more complex biosynthetic pathways that are involved in making substructures that are metal binding. So these chelating groups, they're called. Here you see in this panel B, all of these chelating groups and their biosynthetic pathways. And for each of these chelating group biosynthetic pathway, he made like anti-smash rules to detect the specific combination of enzymes. So to really detect within a complex biosynthetic gene clusters, are there genes present for making these specific substructures as part of the molecules? And yeah, so this actually works pretty well. So you get an F1 score of 0.86. very high precision, meaning that if these subpathways are present, then it's almost always a metallophore. Sometimes you miss things, so the recall is a bit lower because, and I think there was a question earlier, occasionally these genes are elsewhere in the genome. So if these genes are outside the gene cluster, elsewhere in the genome, you might kind of miss out on being able to predict this functionality. There was a previous paper also from UC Berkeley on using transporter genes for prediction of metallophore bioactivity. This works a bit less well, but you can actually also combine these, like looking at the enzymes and the transporters, and together it actually works even a bit better with an F1 of 0.9. Yeah, and this way, Zach was also able to do to get a global sense of, like, across many genomes, like how many of all these gene clusters actually have this kind of functionality. And we're able to actually functionally annotate in this way a pretty substantial fraction of the gene cluster. At the same time, to also expand this to other key types of biological activities, and ideally you would want to do this also with, for example, machine learning methods where you're not dependent on these rigid associations with a specific chemical substructure and a specific biological function. Of course, it would be really cool to do this in a more generalized way. And I do think there is a lot of potential also for AI to help with this. At the same time, and this was highlighted also in this piece in Nature, I think 2 years ago, that there is really a lack of large-scale standardized data to do this well. And this is also something that came up in a workshop that we organized in Leiden a couple of years ago on, like, how we can use AI for, in this case, drug discovery from these natural products. That there's really a need for these high quality, large scale standardized data sets. And I'm happy to say that by now there's really initiatives going on with large scale collaborations to get this going. Also, for example, with large libraries of purified molecules that can be tested across a range of standardized assays to really get better activity data.
So now that Marnix already paved me the way to talk about metabolites and algorithms, I wanted to start by delving a little bit deeper into the human microbiome and the metabolites that they produce. Most of the bacteria that reside in our gut are anaerobic bacteria, so they are the responsible of producing this wide range of metabolites. These metabolites are important for either microbe-host interactions or for microbe-microbe interactions. And depending on the concentrations they are produced, they can have either beneficial effects for our health or detrimental. And this, for example, as an example of this is indole, for example. This is a molecule that is produced in the gut, and in low concentrations, it can have beneficial effects for the immune system. But in high concentrations, this molecule can translocate into the brain, and then it can be at the gut-brain barrier, and then it can disrupt the motor activity of the host. And we have other examples like picrosol, bile acids that can help digest lipid metabolites, and short-chain fatty acids that are well known also for having beneficial effects for the gut by helping tightening the cell junctions and promoting the immune system. But there's still a large black box that we don't know, like, which kind of molecules these microbes are producing. And this is actually not only the case of the human microbiome, but it also applies to other types of microbiomes, like the oral microbiome, the rhizosphere, as Marnix was talking about. So it is very important to know actually who is doing what in these communities, which are the actual mechanisms that are triggering the specific phenotypes that we are interested in. Because if we know we can answer these questions, we will be in a much better position to actually understand and manipulate these communities as desired. And as Marnix was already mentioning before, one of the peculiarities of these metabolites that the gut bacteria produce are known to be encoded in metabolic gene clusters, MGC in short. And these are actually the genes that are close together in the genome, and they are responsible for the regulation, the synthesis, transportation of the metabolite. And this is actually very interesting, but since there was no tool to be able to predict such metabolic gene clusters, we decided to tailor the anti-smash framework that was already working very well for biosynthetic gene clusters for secondary metabolism into gut smash, which Marnix already briefly mentioned before. So we tailored anti-smashed into gut smash to be able to predict specialized primary metabolic gene clusters from anaerobic bacteria, and all the and we actually modified and generate new functionalities to be able to properly annotate and identify this kind of gene clusters. And how gut smash works, it uses logic detection rules. These logic detection rules are PFAM combinations that work together in a way that are relevant because they are the signature of a specific redox mechanism or pathway. So for example, in this case here, we have PFL-like domain and radical SAM domain, or PFL acetate and radical SAM domain. If we find this combination of PFAMs within 20 kbs, this is the signature reaction for the pyruvate to acetate plus formate pathway. And then once we identify the core genes, we extend the cluster on both of the flanks to be able to include the other core biosynthetic gene clusters that might be nearby. And we actually have two different types of rules. One that are known to be specific to known pathways. So this would be, for example, pathways that have the same core enzyme activity, but having like a little bit of flexibility within this pathway so that we can still predict from different organisms. And then we have the general rules that aim to target important redox enzymes in certain contexts that seem to be relevant. And this could be, for example, a flavoenzyme, which is find together with a short chain dehydrogenase. This could be, for example, a novel cluster related to lipid catabolism, but of course, the specific function is yet unknown. So we started from a collection of known pathways that were previously characterized and they were published. So we could have access to the catalytic reaction of this pathway. We knew which were the core enzymes. And we have a collection of these pathways that here you have a representation of these gene clusters. As you can see, they look pretty different from each other. Some of them are very short, some of them are very long. And also in terms of reactions, they are very different. And examples of them are, for example, the 3-methylamine producing gene cluster, the picresol gene cluster, ethanolamine pathway, etc. And so we had for these 3 different pathways that we used to build the known path, the detection rules for these known pathways. And how did we actually build these detection rules? Well, so we started from this collection of known pathways. From there, we could identify the core genes and then discern the core genes from the auxiliary ones. From here, we could also see that some of these core genes were actually repeated between different pathways. So then in those cases, we could generate the phylogenies for these key protein families. And in here, we included protein sequences from different sources. One was from our collection of of specialized primary metabolic gene clusters. Then we also included the protein sequences from the experimentally characterized proteins that were in UniProt. And then we also included a representative proteome from the PFAM database. And from here, we could identify the specific clades that we were interested on. And this helped us to build a specific PHMMs. So we customized the PHMMs to be able to target the enzymes that we were interested on and discern between these ones and, for example, the housekeeping related genes that could be involved in these protein families. And then from here, once we had the PHMMs built, we had a preliminary set of detection rules created. These detection rules were then tested on a collection of taxonomically diverse genomes. And then Marnix, as was mentioning before, we have this this tool in the group called Bigscape that allow us to group each of the predicted gene clusters that were predicted by the same rule. We classified them, and then we run Bigscape on each of these subgroups. And this helped us to classify or group together the gene clusters into gene cluster families. And we manually analyzed the output of the Bigscape to be able to find the taxonomic diversity of the predicted gene clusters, and also if there was any outlier that we can see from the output network, for example. And we also and from these results, we were also able to check, for example, in literature, if what we were predicting was actually known or not known. And from here, we did a couple of, this allowed us to actually refine the detection rules. And we did a couple of back and forth rounds of validation and refinement. And then we had the final version of GATSMASH, which included different types of rules, such as the specific to known pathway rules and the general rules also. So the general GATSMASH workflow then would be to start from a bacterial genome. Then we use the bacterial genome either in GenBank format or FASTA format as input. Then GATSMASH identifies the gene cluster by iterating over the detection rules. Then this gene cluster is well, compared to a database of other gene clusters that we have, and I will talk a little bit more in detail later. And then we annotate the gene cluster, the genes into different functional gene categories. And finally, we build the interactive output that one can see in the results. So to be able to do this comparative genomic analysis, we also constructed 2 different databases. And these databases are known as Known Cluster Blast, which contains the experimentally characterized set of gene clusters. And in here we have for example, this is the output of Known Cluster Blast. Here is the query sequence, and you can see, for example, in here, to which pathways of this database the query sequence looks more like. And you can actually see also, in particular, which genes are the ones that seem to find more more similarity or not. And then we also have the Cluster Blast tool or database that predicts gene clusters that has been, it's a collection of gene clusters that have been predicted from a reference collection using GATSMASH. So in this way, what we can do is to compare the query sequence to a lot larger database of gene clusters. And in this way, we are able to see a little bit the taxonomic distribution of this gene cluster and to see, for example, if this gene cluster is preserved or not, or it's only found in the same genera, for example, or it's a little bit more widespread. Okay, so once we had the tool, we decided to test it out in a data set that contained microbial genomes from healthy donors. And that helped us to profile the metabolic potential of the human microbiota. And for that, we used more than 4,000 genomes. More than 300 genomes came from the HMP dataset. Then we took also the CGR collection genomes that were isolated from stool. And then we also included an extra set of Clostridia genomes to account for their variability. And all of these genomes composed a taxonomically diverse collection of genomes, and these were used as input for GATSMASH. And from here, we were able to pull actually the known gene clusters from the putative gene clusters, thanks to these databases of known cluster blast and this comparative genomic analysis. And here are the results. So for here in the, sorry, I lost my pointer. So here are the different pathways that we have. And here are the different bacteria, the main bacteria that we could find in this reference collection. Here is the total number of pathways, and each of the dot represents the percentage of the bacteria that actually encode for this specific pathway. So if we find a large dot, it means that more than 50% of the bacteria from this genera actually encodes for the pathway. And if the dot is small, it means that less than 50% of the bacteria. And this was just to simplify. And actually, and the colors represent the different types of pathways that we have based on the output metabolites. And for example, what we can see in here is that Clostridium, that is here, sorry. So Clostridium is the genus that actually displays a more metabolically diverse genome. It encodes for 43 different pathways. And then it's followed by the Enterobacteriaceae members, which include the Enterobacter, Escherichia, and Salmonella genuses. And actually, it's interesting because we also can see that despite these are the two genuses that display a remarkable metabolic diversity, there are still many of the pathways where they do not overlap. So that means that they have actually different strategies also to cope and to grow in the human gut. And this is actually important because depending on the resources that they have in the gut, they can actually proliferate more or less, and this shape the composition of the human gut microbiome.
Can I ask a question to that? So for the metabolomic pathways that you were learning on, right, that were your ground truth from known pathways or known databases, have you seen there also a taxonomic bias towards some Enterobacteriaceae that have been better studied in the lab, and which could also explain why you see a high number of pathway matches for them, or Clostridium may be studied well in the lab, so would you basically say that for the other bacteria there are potentially other metabolic pathways, or they can also have a high number but we are not covering them?
Yeah, of course that plays a role also on the identification. I think that the rules how they are being designed, they allow some flexibility, but of course if that differs a lot from what we know, then it's not that we are not going to predict them, but probably this is going to be not classified as known, because based on this comparative genomic analysis, we of course have to set thresholds on, okay, if this is my reference genome, we want this to look more or less similar. So yeah, definitely. but still, like doing in this way, we could still see that, for example, some of the pathways that were originally studied from an organism could be found in another also. And then also here I have a similar plot, but in here, instead of the genuses, I have the specific species. And in here, instead of combining the dots into more than 50% of the isolates having the pathway or not, these are actually row counts. And what we can see in this plot also is that we could find strain-specific patterns of primary metabolism, meaning that some of the, for example, in here, that it's enterocluster albaniensis, I think. For some of the pathways that Gatsmash was predicting, not all the isolates actually encode for this pathway. As you can see in here, some of the dots are very small, implying that not all the isolates were having this pathway. And this is actually very interesting as well, because then it also remarks and highlights the importance of not just considering genuses, but also like strain-specific traits that are important. And yes.
So for this plot map and then the previous one, how much different would it look if you did instead of the I guess phylogenetic tree here, if you just did like a little cluster mapping, right? If you did like a little hierarchical clustering. Because I'm wondering like how much of this is, different, phyla or whatever.
Yeah,
however you
because
want
of the
to describe
taxonomic
this.
classification.
Yeah, like do you have like this specialization occurring at like, the level of, genetic similarity, or is it sort of like, you have different branches and then, further up, and then based on wherever they happen to end up being, they specialize into whatever niche?
I think that's I didn't try this. I think that maybe it would look a little bit different, but also based on this analysis, we could see that, for example, there were some like that actually each of the clades or genuses have very specific functions in the gut. So it's restricted also by genuses. That doesn't imply that then within genuses or strains, we don't have differences also, but I think that the major differences are between taxonomical groups in the higher part of the phylogeny. So yeah. Okay. And then we also decided to analyze a little bit the predicted gene clusters that were generated with, that were differentiated from the known gene clusters. And here, this is a network actually that contains 12,000 different putative gene clusters. That's because we don't know the function associated to them. They have been classified according to the key gene that they encode for. And in this network, what we can see is that there are actually truly novel gene clusters. But there is also, we could also see that, for example, there were gene clusters that were not covered by any specific rule, but that they have been characterized before. So it was not in our initial set of pathways. So then, yeah, we couldn't predict that as a specific pathway. But it's good to see that still with our general rules, we are able to predict these interesting gene clusters. And then, of course, there are the distant homologs of the known gene clusters. That this was Marie's question. That yeah, of course, like if there are some of the gene clusters that look very different to our reference, they were not annotated as known, but they were annotated as putative. And then we also did a more specific analysis on some of these putative gene clusters. And in here, I'm highlighting four of them. So in here on the right-hand side, you have the network where the pathway has been extracted from, and for example, this specific one in here that it's from enterocluster citronae. This one encodes for a flavoenzyme, and the actual neighborhood where you can find this flavoenzyme seems to be very interesting because it's associated with an AMP binding protein and also an acyl-CoA dehydrogenase. And yeah, we wanted to highlight this kind of pathways because we think are interesting, and that these are actually pathways that might be characterized further and might help to uncover novel pathways. And yeah, other examples are, for example, this Doria one, which actually encodes for a pyruvate ferrotoxin, and is actually also close by thiamine pyrophosphate enzyme, which is also very interesting, for example. But this is just to point out some of the pathways that we could find interesting. So in summary from this part, I hope that I showed you that GutSmash can help profile the human microbiome and can help understand who is doing what in this environment. Also that the bacteria are some of the bacteria are very versatile, and we can see strain-specific patterns. And also that the general detection rules are very useful to predict novel gene clusters in bacterial genomes, and can represent interesting candidates to study further. I also wanted to mention that we built the GutSmash web server to be able to just online analyze your genomes without the need of having to install and all the dependencies and the databases and everything. And it's hosted in the gutsmash.bioinformatics.nl webpage. And there's also a bunch of information in these tabs in here. If you click, well, you will find information on how to download, interpret the results, and everything. So now that we know who is doing what in the human microbiome, we were also interested on who was actually contributing to these specific phenotypes. So, and for that, we need to know how abundant are these pathways in the communities, and if these pathways are actually differentially abundant across the samples, because as I was mentioning earlier at the beginning, also like the metabolites have a specific concentration to be able to trigger these phenotypes. So this information is important. And yeah, so if we are able to know if these gene clusters are differentially expressed or not, we will be in a better position to actually understand the role of these metabolites. And I want to highlight that this is actually the tool that I'm about to explain, that is called BigMap. It's not only used for the gut microbiome, but also for other types of microbiomes. So BigMap is a tool to assess gene cluster differential abundance and expression across samples. And we do that by using the predicted gene clusters by GutSmash or any other SMASH tools. And we use this in combination with metagenomic and metatranscriptomic data. And this work, by the way, was done together with Hannah and Koen, that at the time were master students. And BigMap consists of 4 different modules. The first one is called the family module and performs a redundancy filtering on a given set of gene clusters. So we start from a gene cluster collection, and then we run a redundancy filtering using MASH. And then we take a representative using medoids calculation, and then we cluster the representative gene cluster with Bigscape again to be able to get the gene cluster families. Also in case we are using metatranscriptomic data, we also HMM search the PFAM database for 5 different housekeeping scenes to be able to have a baseline of expression levels of these genes that we will later compare to our expression levels. Then we have the download module that downloads the FASTQ files associated to an SRA ID. This step is optional, of course, if the user already have the metagenomic data or metatranscriptomic data, this step can be skipped. And then we map the reads onto the representative gene cluster using bowtie. And yeah, basically what we do is to either align the metagenomic reads or metatranscriptomic reads into the representative gene clusters to be able to have an alignment. And from here, we normalize and further process the map reads to be able to visualize the output. And this is actually how the output looks like. This, for example, this is an example when taking different metagenomic data from a data set that contain Crohn's disease samples and healthy samples. Here in the right, you have the significant gene clusters that were found with BigMap. In the top bar, you have the comparing groups. So in this case, green is for Crohn's disease and yellow for healthy samples. And here we have bars just indicating the log fold chains that were found between the two comparing groups. And then here we have the coverage values of the gene cluster. So the higher is the number, the better covered the gene cluster is by rates. And
sorry, can I ask again?
Yeah.
So do I also get the information by using that tool, for example, which part of the biosynthetic gene cluster has which coverage? So for example, is the transporter upregulated, but the metabolic component is not?
Mm-hmm. Yes. Yeah. So that's not part of the output of BigMap per se, but this is something you can process later with intermediate files that we generate and just visualize it with other tools. This is actually something that I've done for the GATSMASH paper. And I think that I visualized that with the sushi R package, I think it's called. So yeah, definitely then you can easily see which are the parts of the gene cluster that actually have more hits.
Yeah. And this is something that you have seen often, so that, for example, also based on metatranscriptomics data, transporters upregulated, but the rest of the synthetic cluster is not?
We haven't looked at that. Okay. So as I was saying, in this specific case, we can see here that there are two specific gene clusters, one that is known and one that is unknown, that seem to have different abundances in terms of metagenomic reads aligning these gene clusters. They are both, so in here, for example, the more yellow, the more abundant the pathway seems to be, and they seem to be more abundant in the healthy samples than in the Crohn's disease ones. And the coverage of the gene cluster also seem to indicate that they are pretty well covered. So these are good candidates actually to maybe study further. And I also wanted to mention that actually this is this can also help us to prioritize which are the putative gene clusters that we want to study further, because we have here an extra layer of evidence saying that these gene clusters might be playing a role in some of these phenotypes that we are interested in. And this is actually the heat map that would output BigMap for metatranscriptomic data. So here we have used the same data set. So in green, here in green are the Crohn's disease samples, and here the healthy samples. The only difference in here is that we have added this extra column in here, which depicts the expression levels of these 5 housekeeping genes. And that is only to be able to compare our expression levels of our pathways with this housekeeping genes one, for example. And again, in here, we have a gene cluster that is a putative one. that is significantly associated with Crohn's disease, that seems to be more abundant in Crohn's disease. It seems to be related to fatty acids metabolism. And yeah, this is another good example of a gene cluster that can be important for this specific phenotype. Well, now that we know that we have the means to actually know how abundant are these gene clusters, we wanted to know how that would translate into the human gut microbiome. And for that, what we did was to predict the gene clusters, or use the predicted gene clusters from the collection of these 3 different reference collections, in combination with 1,135 metagenomes from healthy samples. This is the data set from the Lifelines deep cohort. And this was actually the input for the big map tool. And in this way, we could assess the abundance and prevalence of the pathways that were predicted by GutSmash. And we also wanted to see if the gene cluster abundance was able to correlate with the metabolite abundance. So for that, we also used big maps results in combination with the MS data of these same participants. These are the results of the pathway prevalence. And so in here in the x-axis, you have all the pathways, and in here in the y-axis, all the prevalence of the pathways, again, classified into the different categories that we defined. And in here, it was nice to see that we were able to capture most of the pathways that were known to be found in healthy samples. We were able to see them in almost 100 in all of the participants. That was very interesting to see. For example, these are these 13 fatty acids producing pathways. All of them seem to be prevalent in all of the samples. And then it was also interesting to see that, for example, the Bay operon that actually transformed the cholic acid into deoxycholic acid, it is known to be encoded in very low abundant bacteria in the gut, that it's Clostridium scindens. And it was interesting to see that also, despite being very low abundant in the gut, we were able to find it in most of the samples also. And then, of course, it was also interesting to see that, for example, for other pathways that are known to be detrimental for the human health, such as the propanediol utilization that, for example, is encoded in Salmonella, or the picrosol synthesis pathway, they were found in a lot less number of samples. And then when we looked at the abundance of these pathways across this data set, we could see that some of the pathways were consistently found across all the samples at high abundance. This is actually the case of P42. This is the 3rd pathway in here. And also, this is the case of all the pathways that actually have a very small box plot and with very short whiskers also. But then we could also find pathways that differ a lot in abundance between individuals. And that's, for example, the threonine to propionate pathway. And this was actually making us question if the metagenomic abundance actually would correlate with the metabolite abundance. Because if we have such differences, maybe we could see those differences also in the metabolite concentration. And this is actually a work that was done together with Lian-Ming Chen. And in here on the top part of the plot, you can see the correlations between metabolite abundance and the pathway abundance. And these 3 lower ones are from fecal content, and the rest are from plasma content. So we also wanted to see if that was actually having an effect on the correlations that we were doing. And yeah, basically we couldn't see a correlation between the metabolite abundance and the pathway abundance, and not in the plasma, neither in the fecal content. Despite in the fecal content, I think that if you compare, because these are for the same pathway, it improves a little bit on the when using the fecal MS data, and that might be because in there the concentration of these metabolites it's higher. And this lack of correlation might be due to different reasons, and one of them can be due to dietary differences, transcriptional regulation differences, diffusion dynamics of the metabolites, secondary fermenters that maybe use some of the end products of some of the pathways and are converted to other ones. So, and from this, we could also see that the importance of using metatranscriptomic data. So that's why we decided to analyze another data set.
Can I ask a question, actually?
Yeah.
In the previous slide, is it that there's only one pathway that's making the metabolite that you're measuring in the plasma or the fecal sample, or could there be multiple pathways that make that metabolite? And so even when one pathway is not abundant, maybe the other pathway is, and that's why you see the metabolite even when the pathway is lowly abundant, and therefore you just will never get a correlation?
No, we actually did all the combinations. So we have indeed different pathways that, for example, produce acetic acid, and we did all the combinations, and we also pulled them all together and then correlate but the results were the same still. So yeah. And yeah, by analyzing, so we wanted to analyze then this IHMP data set that actually contain paired data. So there was 81 samples that have metagenomic data, metabolomic, and metatranscriptomic data. Then there were 271 metatranscriptomic data that were only associated to metabolomic data. And all of these samples actually also come from healthy ulcerative colitis and Crohn's disease samples. So we had different phenotypes in here. But the interesting part is that we were having different types of paired data and metatranscriptomic data also. And this was together with the predicted gene clusters predicted by GutSmash, we used this as input for BigMap, and then with the fecal MS data, we did the same analysis to be able to correlate the gene cluster abundance and expression with the metabolite abundance. And these are the results. So in here, in this first plotting here, we have an overall correlation plot. So here is for the 81 metagenomic samples. The green one is for the 81 metatranscriptomic samples, and the yellow one is the 271 metatranscriptomic samples. And you can see that correlations range between minus 0.3 to almost 0.4. So not very high correlation still. And we also highlighted in here in different colors. I'm not sure if you're able to see the different dots in different colors. And these are actually the pathways that are represented here, just to highlight the few. And if we analyze these specific correlations in particular, because these are actually one of the highest correlations that we get, we see that still the MGC abundance and the expression do not correlate well with metabolite abundance still in general, despite having maybe higher values for the metatranscriptomic value data. Sorry. But actually, when we split it, the data set into the 3 different phenotypes that we could find, we got better results, and actually the correlations increased up to almost 0.60. So yeah, it seems that the phenotypes have actually play a role in correlate on this analysis, right? So it's important to actually split the analysis using these 3 different phenotypes, and also that the expression of some of these pathways seems to be indeed predictive for some of the metabolite abundance, but not all of them, since there's still a lot of them that are close to 0 or very low. So what do we learn from this analysis? Well, metabolic inferences are difficult to make from metagenomic or metatranscriptomic data alone. And this is important because there are tools actually that make such inferences. So they might be very useful, but we have to be cautious also about how we interpret these results. And then they need to better understand the influence of diet, gene regulation, secondary fermenters, and everything to be able to make such predictions more accurately. And then finally, to wrap up, I wanted to highlight a few take-home messages. One of them is that GutSmash can get deeper insights on who can do what, and can be useful to do this metabolic profiling of the human gut bacteria. But we have also actually applied it to different microbiomes, such as sheep rumen, for example, oral microbiome. So it's not just restricted to the gut microbiome, human gut microbiome. Then also that using metagenomic data as input for Big Mac can be very useful to understand how pathways which pathways are actually more abundant, which are not, and the role they play in the phenotypes. Metabolite outcomes are difficult to predict from metagenomic data or metatranscriptomic data alone. And then that I think that integrative omics approaches are the way to go to be able to understand complex ecosystems and infer causation.